Guys, in this video, I'm going to be talking about amino acids and protein structure. So this is the picture of an amino acid, and it has a carboxyl functional group at one end and an amine functional group at the other end. And that is the same for all amino acids. The only thing that changes is the R group. That's the variable group. It's also called the side chain. Now this model here, it shows the different side chains of amino acids. Some are hydrophobic, that is they hate water. Then hydrophilic ones, the ones here in the white side of the chart, they like water. And some are charged. Some are negatively charged, the acidic ones, and some are basic or they are positively charged. Now, in the next model, what I have here is a basic structure of a polypeptide. Polypeptide is many amino acids bound together. So I can open it up like this. And this here is showing the primary structure of proteins. That is the different amino acids that are put together in a sequence. And this sequence is determined by DNA. For example, if this is the hemoglobin protein, then the hemoglobin gene decides which proteins are, which amino acids are going to be put in the chain and in what sequence. So once you have your primary structure, proteins need to fold and get into their proper structure to be able to function. Remember that the shape decides the function, or shape dictates the function. So your protein needs to fold and come to its optimal shape where it can work. And this folding is governed by chemical forces. And we're going to look at a couple of examples. So over here, I have one side chain that is blue in color, the other one that is red. So it is showing an acidic and a basic side chain. And one is negatively charged, one is positively charged. So they're going to be attracted to each other and they will form an ionic bond. So they are going to be stuck and be close to each other. In another region, the green, it stands for the amino acid called cysteine, and it has a sulfur on it. So it is able to bridge with another cysteine to form a disulfide bond. Another example of a bond would be something like hydrogen bond. Different amino acid side chains can form hydrogen bonds with another side chain that may be nearby. So all these interactions keep your protein shape in place. Now, what would happen if the primary structure is changed? That is, if the sequence of amino acid changes. In the example of sickle cell anemia, one amino acid is replaced by another amino acid. Now, if this were to happen, then as the protein is folding, realize that it was supposed to form a cysteine-cysteine bond over here, a sulfhydryl bond, or it was supposed to be like bound together, but now instead of a cysteine over here, we have another amino acid which is not able to interact with this other cysteine. So this protein folding is going to be abnormal. It's not going to be in the proper structure, and which would change the shape of your protein, and it is not going to be a functional protein. So the bottom line here is the primary structure, that is the sequence of amino acids on your protein, is going to decide what the secondary, tertiary, and all other levels of protein folding is, and that will ultimately determine the shape of the protein. So if the primary changes, if you replace one amino acid by another, there is a good chance that it is not going to fold properly and you will not get a functional protein.